Welcome to this week's research seminar. For our speaker this week, it is my great pleasure to introduce Dr. Mohammed El-Kebir, Assistant Professor in the Thomas M. Siebel Center for Computer Science at the University of Illinois, Urbana-Champaign. Dr. El-Kebir received his PhD in computer science from VU University, Amsterdam. After that, he did a postdoctoral training at Brown University and Princeton University before joining UIUC in 2018. Dr. El-Kebir has joint appointments in the Electrical and Computer Engineering, the Institute of Genome Biology, and the National Center for Supercomputing Applications, all in UIUC. His research interest is in the field of bioinformatics and computational biology, and theory and algorithms. More generally, Dr. El-Kebir is developing novel problem statements and corresponding methods to analyze omics data in novel ways, thereby improving scientific discovery. A couple of projects that his lab is currently focusing on includes one, developing methods that enable the estimation of cancer phylogenies from single-cell sequencing data, to developing comprehensive evolutionary models for somatic mutations that occur at varying genomic scales. He received the National Science Foundation CISE Research Initiation Initiative Award in 2019, and this year he received the prestigious Career Award as well from the National Science Foundation to further develop understanding of cancerous tumors by identifying its evolutionary trajectory. I'm really glad to have Dr. El-Kebir today at our Colloquium to share his research work. The title of his talk for today is Combinatorial Algorithms for Tumor Phylogenetics. Welcome again. Thanks for being here, and the Zoom floor is all yours.
Thank you so much, for the introduction and for the kind invitation. So it's my pleasure to give a talk about my research. So we're going to be talking about cancer today, so let me start by just giving a very brief overview. So by the way, if there's any questions, so we're a small audience, so just feel free to speak up or type in the chat. I'm monitoring the chat as well. Okay, so let's take a look at cancer evolution. So I'm just going to go back here. So you can trace back a tumor to the very first cell that acquired a somatic mutation. So somatic mutation is a mutation that that is new. So a mutation that you did not inherit from your parents. Because of this somatic mutation, this tumor cell was fitter than surrounding cells. And this led to something that's called a clonal expansion. So it divided faster than surrounding cells. So you see more, descendant cells with this initial light blue mutation. And descendants of this very first tumor cell, they can acquire new mutations themselves, leading to further clonal expansions. And what you get in the end is this heterogeneous tumor. So we call this phenomenon intratumor heterogeneity. So there's within one tumor, you have multiple subpopulations of cells that each have distinct sets of mutations. And this is evolution. So we can represent this process by a phylogenetic tree. And this is really the key object that we try to identify or reason about in cancer phylogenetics. So let me just give you a brief motivation. So why do we care about this? So there's two reasons. So let me start with the basic science reasons. So even though every tumor is the result of a separate evolutionary process, you can still compare tumors from different patients and try to identify common patterns of tumor evolution. And in order to do that, you actually need to reconstruct tumor phylogenies in individual tumors accurately. The other thing that is very relevant that you can study with tumor phylogenies is the process of metastasis. So, most cancer patients, 90% of cancer patients, they die because of metastasis, not because of the primary tumor. So understanding this process is extremely important. And, all metastases, including the primary tumor, they're all related by the same phylogenetic tree. So if you reconstruct this tree, you can actually start thinking about patterns of spread between locations. And this helps you understand how the tumors spread from one side to another side, and if there's any recurring patterns across patients. And finally, this is ultimately where the field is headed, is can we understanding the evolution of a tumor, can we actually think about how we could alter the evolutionary trajectory of the tumor by identifying treatment targets, so mutations that occur in different sets of cells. So you could basically alternate treating one set of clones versus another set of clones, and in that way you can actually kind of like control the tumor. So this is evolutionary steering. We're not there yet, but that's kind of like we're something that we ultimately want to achieve. Yes, so there's a question in the chat about viral origins. Yes, so there are so essentially these somatic mutations that can result due to different reasons. There can be environmental factors, but there are certain tumors that have where somatic mutations result due to viral origins. So that's true. Yes. So in any case, all these downstream analysis, they critically rely on accurate tumor phylogeny inference, and this is actually not an easy thing to do, because we're trying to make inferences about the past from data at the present time. So this is a challenge in phylogenetics. So not unique to cancer, just in general phylogenetics, but in cancer, there's actually an additional challenge, and this has to do with our sequencing data. So most cancer sequencing studies, they do bulk DNA sequencing. So what this actually means is you take two tissue samples. You take a tissue sample from the tumor, and you take a tissue sample outside of the tumor. So that's called a matched normal. And then you sequence both samples and you align them to the human reference genome. So typically, this is next generation sequencing. So we get short reads. And as a first step, what you do is you can compare it to alignments. And this allows you to identify mutations that are unique to the tumor sample that you don't see in a normal. So this gives you a sense of what are the somatic mutations that we see. Now the challenge is that this tumor sample is not homogeneous. So there's intertumor heterogeneity. So there are multiple subpopulations of cells with different sets of mutations. So this combined with like the short reads that we're getting makes it really hard to understand patterns of co-occurrence or mutual exclusivity among mutations. So when I look at this orange mutation and I compare it to the green mutation, I don't know if they co-occur in the same cells or not. So that's basically a key challenge that we have here. So we have this mixture data that we need to deconvolve. So how do you, on top of like trying to reconstruct the past from data at the present time, we actually have data at the present time from mixed bulk samples. So this is different from standard species phylogenetics, where you don't have samples that are mixtures of different species. In this case, we actually have samples that are mixtures of different clones. So that makes it really challenging. And I'm going to be talking about this today. And I'll basically first dig in the theory. So I'll just introduce a problem statement for solving this problem, or that tries to model this problem. I'll show you some simulation results about this problem statement. And we're going to be focusing on multiple optimal solutions, so this non-uniqueness of solutions. And then finally, we're going to look at, can we actually sample solutions from this space uniformly at random? And are there ways to summarize the solution space? And I'll end with one or two applications, downstream applications. So let's get started with the background and theory. So I explained this already. So instead of just taking one tumor sample, you can actually take multiple tumor samples from a cancer patient. So I'm indicating them here with S1, S2, and S3. And in addition, we've also taken a matched normal sample. And these 4 samples in this toy example, they've all been sequenced, and we aligned the resulting reads. And then we basically compared each tumor sample to the matched normal, and this allowed us to identify, end positions in the genome that are mutated. So for now, we're just gonna look at point mutations. So for instance, this 0.8 frequency of 0.8 in sample 3 of the light blue mutation. What that actually corresponds to is in your alignment, there's a position, a locus, where, in this case, we had 5 reads, and 4 out of 5 reads had the mutation, had the variant allele. So what this actually means is, like in the normal, in the matched normal, there's only C. We don't see a G. Okay, so this G is the variant allele, and the variant allele frequency is 0.8. So we measure this variant allele frequency for N mutations in M samples. And we can represent our input with this frequency matrix. So that's our input. Now this input is actually the result of mixtures of unknown leaves, of an unknown tree, in unknown proportions. And what we're trying to do is we're trying to, given the frequencies, we want to find, the phylogeny T, that would be this over here, and the proportions of the leaves of the tree that resulted in every sample. So let's take a look here. So for instance, this sample 1 over here is very simple. So 0.8, so 80% of cells, they look like this. So that's why we see a frequency of 0.8 of like the light blue, green, and purple mutation. So the remainder, so the 20% that is remaining, that actually corresponds to normal cells. So whenever you take a tissue sample from a tumor, you will always get normal cells in there, but we don't include them in the tree. So sample 3 is more complicated. So here we actually have a mixture of 3 different clones, 3 different leaves in different proportions. But that's the relationship. So we have this tree, and then the proportions that describe how the leaves of the tree were mixed, and that yields our input. But we're going for the inverse problem. So given the frequencies F, can we reconstruct, find this output is tree T, and the proportion is U. Good. So that's what we're trying to do here at a high level. And in order to do this, we actually need to reason about the tree T. So more specifically, we need to impose an evolutionary model on the tree T. So the evolutionary model we're going to look at is the most constrained thing possible. And the reason why we do that is because it actually leads to it makes the problem, you get a good handle on the problem. So we're going to make the infinite size assumption. So the infinite size assumption, what it states is that a mutation occurs only once on the tree, and once a mutation happened, it won't be lost. So what this actually means is you can see it in this tree over here. So we had 6 mutations. Okay. So the light blue mutation happened here on the first node. Okay. And the other, what that means is we won't see it elsewhere appearing again in the tree, but we also won't lose it. So every node descendant of the root node will have the light blue mutation. So this is also known as the perfect phylogeny. So this tree is also known as a perfect phylogeny tree. And the cool thing is we can actually represent this tree with a binary matrix. And we're going to make one little change here to classical perfect phylogeny. So we're actually requiring that every edge is labeled by exactly one mutation. And if you do that, you get this beautiful square matrix here, this beautiful square binary matrix. So the columns correspond to the mutations, the n mutations that we have, and the rows correspond to the nodes of the tree. And basically, it tells you for every node, what are the mutations that are present in that node. So that's what you see over here. So this is this binary matrix B that we have, and there's a one-to-one correspondence between trees, perfect phylogeny trees, and these binary matrices B. Now, given this binary matrix, what we're doing actually is we can model the mixture of the nodes of the tree with this matrix U. So rows correspond to samples, and columns correspond to to the nodes of the tree. So that's the same. And entries in this matrix, they are non-negative, and each row sums to at most 1. So as I said, the remainder, that will be the proportion of normal cells. Now, what we can do is, the forward problem, computing the frequencies given U and B is very easy. It's just F equals U times B. It's just matrix multiplication that we do. What we have here is the inverse problem. So given F, we want to find U and B. So let me just summarize. So what I did here is, I kind of like got rid of the tree a little bit. So I'm representing this tree as a binary matrix, and I'm using the factor as a one-to-one correspondence between trees under the infinite size assumption and binary matrices. So these are not just, any binary matrix, they're actually constrained. So we'll talk about those constraints soon. So this becomes a non-negative matrix factorization problem, but with a couple of changes. So B has to be a binary matrix, but it actually has to be not just only a binary matrix, it has to be a binary matrix that corresponds to a tree. And U has to have non-negative entries, and every row must sum to at most 1. So those are the two key changes that we have here. Good. So that's the problem. So how do we solve this problem? And essentially what I'm showing you here, like this problem, a lot of methods in cancer phylogenetics that operate from bulk sequencing data, they use this model. And so on the previous slide, I was saying, error-free data. I'm essentially assuming, I'm trying to get a very clean model. We're going to incorporate sequencing error later. But for now, just to understand the underlying computational problem, let's just say that these frequencies are error-free. They actually represent really the mutation frequencies in the population rather than a sample that you took. But this problem that I showed you here, this is pretty much what underlies every method in the field that tries to do tumor phylogeny inference from bulk sequencing data. There's variations, obviously, that talk about how do you model sequencing errors? Can we do something likelihood based? But under the hood, this is really what's going on. So let's talk a little bit about how to solve this problem. So I'm gonna make it a little bit simpler. So let's just say, so in the problem we were only given F and we wanted to find B and U, but let's just say, we're given a tree as well, or equivalently a binary matrix B. Is there, how do we find the corresponding matrix U? Now so I promised that I would talk about this binary matrix B and T. So you can actually reconstruct a tree directly when you look at the binary matrix B, because the root node corresponds to a row that has just a single mutation, right? And then everything with Hamming distance 1, that's an edge. So if I see basically, if I compare these first two rows, I see there's one new mutation being added. So that would tell me it's an edge of the tree, and the new mutation that's introduced, that's basically the mutation that's going to label the edge. Okay. So the set of binary matrices that we're actually interested in are those binary matrices where there's exactly one row that has a single entry, and every remaining row has a unique parent, which means there is another row with one fewer entry, one entry less. So what you can do is you can actually arrange the mutations in such a way that they appear on a diagonal. And there's a very clean structure to this. So the properties I was describing about, there's a single edge and this Hamming distance 1, what it actually means is that this matrix is lower triangular, which means, so it's a square matrix. So that also means it's invertible. So, given, F and B, U is actually uniquely determined. U is just, F times B inverse. Now, what we showed is this B inverse. You actually don't need to compute B inverse. We have a closed form equation on U that is much simpler, and it's actually very insightful what's going on. So let me just explain what's going on here. So if we try to compute an entry U in the tree. So for instance, let's talk about this U over here. So I'm trying to compute this entry U for instance, let's just say this sample. So what you do is you look at the frequency of the mutation that was introduced. So that will be the the green mutation over here, and you subtract the frequencies of the children. So in other words, the frequency, what it actually tells you is it's the mass of the subtree rooted at a node where that mutation was introduced. And if I want to get the mass of that individual node, I need to subtract the masses of the subtrees of the children. So that's really the relationship between F and U. So given that, we can actually we can compute U directly. Now there's a couple of interesting things that are happening. So let's take a look at this example. So in this example, we're looking at this star tree. So these are, by the way, rooted trees. So there's direction here. So we have this star tree. And if I compute U using this equation, what I'm doing is I'm doing 0.15 minus 0.1 minus 0.06, so that leads to a negative entry. So this actually tells me is that our data does not support this tree. So that's pretty cool. So we actually we know that not every tree is possible, right? So this gives rise to a brute force algorithm immediately. So what you can do is you can just brute force all possible trees, or n. So trees with n nodes, and where the nodes are labeled uniquely by a mutation. And then try, and compute the corresponding matrix U, and then you discard those trees where there was a negative entry. So this is not going to scale because the number of trees that you have with n mutations is n to the n minus 1. So that's not a feasible strategy. So what is it what we did here? Let me just move on. So we were actually trying to, reason a little bit about, the solutions. So what are trees, given F, what are trees that would not incur or induce a matrix U with negative entries? So, which trees would actually give you an admissible U? So if you look at this equation over here, so obviously what you want is you want this left. So every frequency is between 0 and 1, right? They're variantly frequencies. So what you want is that this frequency here, the red box, is greater or equal to the sum of the children, the sum of the frequencies for the children. And that's precisely what is needed. So we actually prove that this is a necessary and sufficient condition to solutions. Now, what you can do is something really cool. So you can take this condition and you can relax it. So instead of, so the frequencies are all non-negative. They're greater or equal to 0. So I can make this a little bit weaker. So if I drop the sum, so if this holds, then this also has to hold. So every individual child, the frequency of every individual child cannot exceed the frequency of the parent. So this is a necessary condition. It's no longer sufficient because we are not looking at all the children. We're just looking at one child. But this ancestry condition is actually super useful because it allows you to construct a graph. So what we're going to do is we're going to take our frequency matrix F, the nodes of the graph, the directed graph, they will correspond to columns or mutations. And there's going to be a directed edge, if and only if the frequency of the parent is greater or equal to the frequency of the child across all samples. So we have an edge between light blue and green because the frequency of the parent 0.8 is greater than 0.6, 0.7 is greater than 0.6, etc. So that holds for all samples. So this gives you this graph, and we call this the ancestry graph. So the ancestry graph you can construct directly from your frequencies, and solutions to our problem correspond to spanning trees in this graph. Not just any spanning tree, it has to be a spanning tree that actually satisfies this sum condition. So it has to be a spanning tree. So if I look at this red tree over here, if I look at the frequencies of the children, so since there are edges, every individual child will have a frequency that does not exceed a parent by definition. But we're requiring more. So we want the sum of the children, the frequencies of the children, they cannot exceed a parent. This 0.6 plus 0.1 cannot be greater than 0.8. That holds for every sample. And every node of the spanning tree, then we have a valid solution. So that's something that we prove. So solutions correspond to spanning trees of this graph that satisfy the sum condition. Now, finding spanning trees or finding a spanning tree in a graph, that's a simple problem. But we actually proved that finding a constrained spanning tree that satisfies the sum condition, or deciding whether there exists one, is NP complete. And it's actually, it's NP complete even if the number of samples is 2. So if you have 2 rows in our matrix. So that's basically what's going on here. So that's the underlying problem that we're looking at. Any questions so far? Okay. So deciding whether there exists a solution is hard. It's NP complete. But there's something interesting going on, which is actually not surprising because we're looking at mixtures. So the problem is underdetermined. So from the same frequency matrix F, we can actually infer many different trees that are all admissible. Yeah, so there's a very good remark about the infinite sites assumption. Yes, it is very restrictive. That's correct. So let me just repeat the assumption. So the assumption is that every mutation happens exactly once in the tumor and is never subsequently lost. So let me break that down. So there are actually it's 2 assumptions. So this happens only once. That's reasonable for mutations that are not that don't confer a selective advantage. So in cancer, mutations that confer a selective advantage, those are driver mutations, and they're just a small number. There's only a few driver mutations compared to a lot of passenger mutations. So when we look at tumors in practice, we're talking about 5 to 10 drivers compared to 5,000 passenger mutations. So that's the gain, the single gain. The loss part, that is actually a little bit more tricky. So loss can happen because of copy number aberrations, where a genomic segment, like a chromosome arm, is actually deleted later on. And this is something that my lab and also others in the field have looked at. So you can extend this basic model, but it's still very important to understand this model. So if you can't do anything reasonable here, then there is no hope of doing something reasonable on more complicated evolutionary models. So this is still a very valid model to study and understand. And for simple tumors, like pediatric tumors or liquid tumors, you don't see copy number aberrations, and your drivers, so you can just exclude them and then just focus on the passenger mutations, and this would actually be very reasonable to do. Copy number variations. Yeah, you could view them as so copy number variations where you would have a gain in amplification that would lead to a copy of a large genomic region. It can be in tandem, so it will be repeated, but this is way larger scale than small repetitive sequences. So we're talking about they can be focal, so you can have a single gene like about 50 kb. But oftentimes we're talking about chromosome arm level events. And it's not just gains, but also losses. And the losses are the ones that are problematic with our assumption. Good. Okay, so let me go back to this slide. So there are multiple admissible solutions from the same input data. So in this very specific example, this mutation 5, we can place it as a child of 3, we can place it as a child of 2, and we can place it as a child of 1. And they all lead to matrices U. You will get different matrices U. You get a U1, a U2, and a U3 that are different, but they're all fine. They all have non-negative entries. So we were interested in 2 questions. So question number 1 is, can we determine the number of solutions? And question number 2 is, if this number is really large, if it's so large that we cannot actually enumerate all of them, can we actually get samples uniformly at random? That's the next best thing that you would want to do. So those are the 2 questions that we focused on next in our research. So let's move on here. Yeah, I don't think I need to be telling you this. So just very quickly, so we looked at that decision problem. So that was about, F equals UB subject to the sum condition. That's just the decision problem. So, is there a solution? And we can actually turn this into a optimization problem. So if we would impose like a likelihood model on top of it. So we're trying to this F that we have here, that's not really the population F. So we're trying to find another F prime that, and we can score the difference with a likelihood model, to capture sequencing errors, and then on this F prime, we actually do the inference. For the F prime, we want to find this U and B. So that turns this into an optimization problem. And the things that I'm interested in is, can we count the number of solutions, and can we sample uniformly from the solution space? Okay, so let's talk a little bit about the counting problem first. So in the counting problem, we want to count the number of solutions. Okay, and what we showed, or what we, so the decision problem was NP complete, and this counting problem, what we actually proved is that it's sharp P complete. So it's actually really hard to count the number of solutions, and what we also showed is that it's hard to approximately or almost uniformly sample from the solution space. So this was a paper written by my PhD student, so we were just investigating like the theoretical complexity of this problem, and it has actually practical implications that we'll discuss next, because there's a lot of algorithms that are based on MCMC, Markov chain Monte Carlo, that try to sample from the solution space, and they will have convergence issues because of this, these theoretical results. So let's talk about that actually. but before talking about how this non-uniqueness affects current methods, I want to discuss reasons for why we have this non-uniqueness, and how we can actually reduce it. So very briefly, so this what contributes to non-uniqueness. So we did simulations here, and so we basically had a model that would allow us to simulate trees, and then we computed we drew a matrix U, and we generated our frequencies F, and then we just enumerated all possible solutions. And that's what I'm showing here on the y-axis, like how many solutions do we have. And I'm showing this as a function of the number of mutations that we simulated, and also the number of bulk samples that you've taken. So those are the colors. So when we look at this plot, so the first thing that we see is that the number of solutions actually grows exponentially with the number of mutations, but it's actually counteracted by the number of samples that you take. So that's actually super important. And in particular, with a single sample, it's really bad. And with a single sample, a lot of solutions are actually possible. In particular, one solution that will always be possible is just the linear phylogeny, where you order the mutations from largest frequency to smallest frequency. So only with more samples can you actually start reducing the solution space. So that's a very important thing to note. And yeah, I hinted at this already. So as you increase the number of samples, you actually get more branching. Like you can actually identify more branching, uniquely. So that's the effect of having more samples. And the next thing that we were trying to do is can we actually reduce this non-uniqueness if we would, for instance, switch sequencing technology? So most common used sequencing technology is next generation sequencing, where you get short reads, and we were wondering if you would replace this with longer reads, what would happen with the solution space? And what we saw is that if your reads get longer and longer, and as such they're actually able to cover multiple SMVs with the same read, that's actually an important signal that you can leverage to your advantage. Because if you have two mutations covered by the same read, so for instance, if I see that this light blue and the purple mutation are covered by the same read, then I know that this purple mutation and the light blue, they must be on the same branch of the tree. And that actually is a powerful constraint that allows you to reduce the solution space. And taking this one step further, if you would combine bulk plus single cell sequencing, you can actually, because if you do single cell sequencing, if it was error-free, so for instance, if you see a cell that has purple, green, and light blue, then you know that they must be, a connected path of your tree. And that's a very powerful constraint that you can add, and this will reduce the number of solutions by quite a bit. And this is something that happens in practice. So a lot of studies actually use bulk plus single cell combined. Good. And the next thing I want to mention very briefly is, so remember how I told you that solutions are spanning trees that satisfy this sum condition. So if we drop the sum condition, we get an upper bound on the number of solutions. So that's what I'm showing here on this right plot. So this is just the number of spanning trees that we have, and this is the number of spanning trees that satisfy the sum condition. So if you look at a ratio, it's actually very interesting. So if you look at this ratio, you can see that this gap that you have between just spanning trees versus spanning trees that satisfy the sum condition, it just keeps on growing, and it's actually something that you cannot bound. And this has important implications because spanning trees, counting them is actually very easy. You can do it in polynomial time, and it's also very, very easy to sample spanning trees uniformly at random. So if this gap was actually bounded by a constant, then what you could do is you could have done like a simple rejection sampling scheme where you would just sample spanning trees, and then you would check for every sampled spanning tree whether it's actually meeting the sum condition or not. So, and you would reject if it does not, and if it does, then you have a solution. But as you can see here, this is not going to really work in practice. So this is essentially the success probability that you would have for such a scheme, and it rapidly decreases. Now, the next thing that I want to mention is I want to look at these two methods that do MCMC to sample from the solution space. There's a very good question, Mathieu. If longer sequences are used, the complexity of the counting problem. No, they won't, because you can still have, so there will be mutations. Let me see how I can say this. So, even if the sequence was as long as possible, you would still have problems with mutations that occur across different chromosomes, and you would have mixtures on those. So that wouldn't change. Well, that would make it a constant. I have to think about it. That's actually interesting. But in practice, the read length that we see for long read sequencing, that's not long enough to cover an entire chromosome. But that's a very interesting question. Thanks. Okay, so going back to this, these methods that do MCMC. So for RWGS, it basically, so what we did here is we just took 10,000 samples. So then we generated 10,000 trees using PhyloGS. And we did this on a data set where we actually had enumerated already the trees in our solution space. So in the solution space, we knew there were 297 trees. And PhyloGS just returns 67 out of those 297 trees. So there are 10,000 trees, and if you look at and a lot of them will be will be copies of other trees. There's a lot of repetitions, but if you make them all unique, you see that there's only 67 out of the 297 that are covered. So there's this non-uniformity that they suffer from. So they're not really sampling the solution space properly, and this is problematic because these other trees, they could have been actually the underlying ground truth tree, and you should actually study them. So you don't want to be getting a biased sample and then draw conclusions based on that. And this problem is even more pronounced for this other method, Canopy, which also does MCMC, but it's much slower. And for this instance, this was actually small enough where we could do a rejection sampling, so the same procedure I described. So that works, that works really well. But the problem with rejection sampling is, as I showed, is that this gap between spanning trees and spanning trees that satisfy the sum condition, this keeps on increasing. So it will break down pretty soon as the number of mutations increases. So in practice, we're actually, I mentioned a number of 5,000 mutations. So we're not actually able to get trees with 5,000 mutations because the data is not precise enough. So we only sequence up to a certain coverage. So what we do in practice is we actually cluster mutations into groups and then do tree inference on those groups of mutations, on clusters of mutations. And in practice, we can, we're actually in this regime right now, 15 to 20 for more complex tumors. Good. So what am I showing here? Yeah. So this problem is actually so this was for a single bulk sample. So if you take more bulk samples, the number of trees in the solution space reduces. But these MCMC methods still suffer from they're not returning the trees in the solution space. So for instance, Canopy here only sampled 1 out of the 6 ground truth trees, and oftentimes it actually samples something that does not even occur in the solution space. So they're having really, they're having issues with convergence here. So the next thing I want to touch on is like, what is our solution to this problem? So, how do we approach this? Like, can we do something better? And so let me just show you the results immediately, and I'll tell you how we did this. So we were actually able to, sample, so the same instance I was showing you, that's here in panel A. So we got like, this distribution in about 3 seconds, and for more complicated samples, it scales really well. And we used a nice reduction here. So what we did is we used this recent work by Maciej Wardy from Rice University, where they've developed an almost uniform sample for satisfiability. So satisfiability, just to repeat or recall, is, so we're given a Boolean formula, a propositional formula, and we're trying to find a truth assignment. So we want to basically decide for every variable whether it's true or false, such that the whole formula is satisfied. And what Vardy and colleagues did is they have this almost uniform sample for satisfiability. And in order to make use of this, we actually had to reduce our problem to satisfiability. And that was a very hard exercise to do. So we started with a simple case. So we were thinking about, what if there's no error in F? And let's just also assume there is no repeated column. So that means that the graph is a DAG. You can actually represent, spanning trees very easily. So you can basically represent whether an H is part of the solution or not. And that's very easy to represent. So there's just a single root node, and you want to maintain connectivity. So that's precise. Well, you want to maintain connectivity, and on top of that, so for every mutation, Q, you want to have a unique parent. So you can only have a single edge. So for every Q that is not equal to the root node, you want to have a single edge. So those are the two things that you can model. And this is very easy. This is something you can do easily with satisfiability. Now, the tricky thing is this sum condition. So just to remind you, so it's essentially that the frequency of the parent is greater or equal to the sum of the frequencies child of I. So this is the condition. So we need to do arithmetic. And what we did is, we first turned these frequencies into integers. So we discretized them, up to a precision that is specified by the user. And then we actually had to represent addition and this inequality over here. So that's, so that took some engineering, and we designed circuits for doing that. So I'm going to skip over that. And this actually allowed us to, to achieve these results over here. And I don't have slides for this, but what we did on top of that is we actually also were able to look at a likelihood-based version of this, where we model read errors or variant reads as a binomial distribution. So this is work in progress. So we're still finishing this up. Good. So let me see. So 152. So I'm just going to touch on this very quickly, and then I'll wrap up. So what can we do when we actually have all these trees that we sample? Like, how do we summarize them? So just to illustrate what I mean by that, so this comes from a New England Journal of Medicine paper, where for this one patient, they actually inferred 17 trees. So the question that we asked is like, how do you summarize these trees? So that we can actually remove inference errors and identify dependencies among mutations. So are there common patterns among the trees that we have for this patient? Because only one tree is true. That's really what's going on. So there's a couple of ways of approaching this problem. So you could just, represent this as a graph. So you take the union of all the edges, and for every edge, you can basically count in how many trees do I see this edge. So this is not great because it actually does not capture patterns of mutual exclusivity. So trees that had this edge, V1 to V7. So if you had this edge, this one over here, you don't have the edge V4 to V10, but you don't see that here. So that's challenging. You don't want to be doing this. So you want to use consensus trees, and this is something that was done by Usper and colleagues, where they basically were going for a single consensus tree, and they showed that this actually under some distance function, it corresponds to finding the maximum weight spanning tree in this graph. And the challenge with this summary is that it can be an inaccurate summary of the single tree that you get if your solution space is diverse. So what we did in this work, we formulated a problem of finding multiple consensus trees. So this is an ISMB 2019 paper where we were seeking to, we were given these trees for one patient, and we were trying to partition them into K clusters and find a consensus tree for each cluster. And this becomes a very clean problem, and I don't have time to discuss this too much, so I'm just going to skip to the results very quickly here. Yeah, so for this patient that we start with, that I showed at the very beginning, so using our approach, you can actually find two clusters, and this cluster has 10 trees, and this cluster has 7 trees. And they actually capture different properties. So for example, this tree over here has this edge V1 to V7. So these are, mutation clusters, okay? And, this tree over here has the edge V4 to V10, and they were actually mutually exclusive. So you don't want to be seeing them together, and we were able to recover this pattern. So that's actually pretty cool here. Cluster and consensus. So a cluster is, so we have 17 trees. So T1, T2, all the way up to 17. And a cluster is just, a partition of the trees into clusters. And then for each cluster, so here we basically took 10 trees in this one. Well, not in this order specifically, and then 7 in this one. And we had a consensus for each cluster. And that's what I'm showing here in dark blue, and this one in light blue. So they mean two different things. So each cluster has a unique consensus tree. Yeah. Good. So that's what this approach is doing. And so you can actually reduce. So even if your solution space is pretty large, you can use this approach to get a small number of consensus trees, and then you can use those consensus trees in downstream analysis. And I won't have time to talk about the downstream application, but I'll just skip to the end and take questions. So let me just get to my summary slide here. So when I started, the point I made is that downstream analysis in cancer genomics, they critically rely on accurate tumor phylogeny inference. And, we discussed a very widely used evolutionary model, the infinite size assumption, and we looked at theory and background of that. We looked at factors that contribute to non-uniqueness and the impact of that, and then we saw an algorithm that actually outperforms current MCMC approaches and gives you a good summary of the solution space, a good sampling of the solution space, almost uniformly at random. And we saw a way to summarize that, and we didn't get into this today. But this is essentially like a pipeline. So whatever you sample, you can summarize, and then you can do a downstream application where you evaluate a tree in different ways, try to interpret it. And I'd like to thank the students who actually did all this work. So Yan is still a PhD student in my lab. So Sarah, she just graduated. Yeah, I'd also like to acknowledge the funding agencies for this work. Okay, thank you so much. Happy to take any questions.
All right, thank you so much. Okay, it's open to questions. So maybe I'll quickly, so everything that you infer, how is that going to help with in the cancer, in the clinical side of things? What is this going to infer, right?
Yeah, that's a good question. So we're still very much upstream here, like on the research side. So we're trying to reconstruct evolutionary histories of tumor. But once that problem is done, or if you're able to do it accurately, that's where things really become interesting, because then you can do things like evolutionary subtyping. So you have a cancer in the clinic, and right now in the clinic, they just look at mutations without looking at the evolution of those mutations. And but if you actually looked at a tree, you can subtype it based on other trees that you've seen, and maybe you already know how they respond to therapy, and you can guide your therapy based on that. So that would basically change, treatment. So it should not be based on like a static, flat list of mutations, but it should be about, how do these mutations actually correspond or co-locate within clones, and what's going to happen in the future. So that's ultimately what should become possible once we're able to do tree inference accurately.
There's a question in the chat. How do you contrast this model with clinic assays?
Could you elaborate on clinical assays? Assays to do what?
Professor?
Yes.
Yes,
please. Yeah.
If you want to check if your model has validity, have you worked with the clinicians to know how it works?
You mean like the infinite size assumption and things like that, the evolutionary model? Yes, okay, let me just back up. So the cancer sequencing studies that we look at, so there is a couple of very big ones, like the Cancer Genome Atlas and the International Cancer Genomics Consortium. There are clinicians involved in them. So this is like, worldwide, there are clinicians and computational biologists. We develop these models, together with, biologists and clinicians. Now, the problem is this is complicated. So your models need to allow for accurate or tractable inference. So you have to make assumptions, and we are aware what these assumptions break down. And even like when you write a paper, you can actually test, if the assumptions are violated, how accurate is your inference, and we always do that.
Thank you.
Yeah.
Any more questions? So you mentioned, you talked about Philo, WGS, and Canopy. So are there programs that generates trees, and then you're using those trees in your experiments?
Yes, so they generate trees, and so in bullet 5, we look at trees from those methods. But here, like the things that I was showing today, is I was comparing their trees against my trees, or the ground truth trees.
Oh, okay.
Yeah.
Okay. So the sequencing data, you somewhere along the way you mentioned single cell sequencing. So is that so? Does it matter if it is resequencing, or is it are you just focusing on single cell sequence data? I was unclear on the data that you are putting.
Okay. What I showed today is bulk DNA sequences. It's not single cell. This is where you sequence millions of cells simultaneously, and this is where this problem arises of mixed samples and frequencies, mutation frequencies that you need to deconvolve. So with single cell sequencing, you still have non-uniqueness, and the reason is because it's not completely error-free. So there are high, and the biggest problem is dropout. So the absence of a mutation in a cell, if you don't see it in a single cell sequencing experiment, does not mean that the mutation was really absent. It could also have been a dropout, so a false negative. And we have really high rates of false negatives, about 25%. And there are different ways of correcting them. And yeah, you can often find multiple optimal and near optimal solutions that you cannot really disambiguate. So that would still lead to a large solution space. But like the frequency, like the F equals UB, it does not apply there.
Thank you.
Thanks.
Any other questions? That's a lot of computational complexity that you went through, so. Okay, I think if there are no more questions, let's thank Mohammed one more time for the great talk.
Thank you so much.
Okay.
All right. Thank you for the invitation.
Thanks. Yeah, thanks, Mohammed. Maybe we can have a follow-up chat and then
sounds good.
Actually talk about other things.
Yes, I'll stay on the call.
Okay.
Thank you.
So the tools, are they available that we could potentially try, or?
Yes. I should have included actually a link to my webpage. So yes, so they're all on GitHub, and yeah. Yeah. So yeah, typically, yeah. So there's different types of tools. So like Phylogenetics, Canopy, they're MCMC based. I think Phylogenetics is like a Python/C++ thing. Canopy is like R. What we do in the lab a lot is like ILPs, like integer linear programming. So where you would need Gurobi or Cplex, but you can get it for free, as an academic institution.
So the data set, do you use primarily the one from the Cancer Genome Atlas?
I use those, but I also so the really cool thing is, like, a lot of all these publications where they analyze like new data, they have to make the data available, and that's where I get it from.
Okay.
Yeah, and then often you can like reanalyze it like using your algorithm, and sometimes you find like different conclusions.
So the NSF project, is that going to be a continuation of this work, or is that something that's going to be focused on?
I wrote it about single cell sequencing, so that was something I did not really discuss today.
Okay.
Yeah. Yeah, and the other thing I did not discuss today, I hinted at it briefly, is, well, actually, let me share my screen very quickly.
Yeah, I mean, if you can, maybe we still have a few students, so maybe you can, if
Sure.
you have time, if you have
Yes, yes,
time.
I have some time.
I'm interested in the applications.
Okay. Well, okay, good, very good. I can do that. But I think this slide is actually very insightful. So, I was talking about point mutations or single nucleotide variants. So this would be, one letter changes base. So this can be pretty, it could change the amino acid, or worse, it could actually lead to a premature stop codon. So this is what happens in TP53, for instance, like if you get a premature stop codon or a truncated mutation, so the protein doesn't form or doesn't fully form. But there's more. So you also have like small insertions and deletions, but it goes up to like copy number aberrations. That's this whole scale where you have like large genomic regions or like chromosome arms that are amplified or deleted. And like some tumors actually also, had a whole genome duplication where, instead of 2 copies, you have 4 copies, and then, you can do whatever you want. It's a mess. But that's also a lot of tumors are like that, where there was a whole genome duplication like a decade ago. It's hard. Like there's a lot of research to time them. Like there was this one paper where they timed it like a decade to 2 decades.
But are you focusing mainly on the single nucleotide variations?
Initially, but like the career and everything that I'm doing, like we're looking at copy number aberrations as well, and the interplay between them. That's the tricky thing. Like if you have, this is what I mentioned. So if you have an SMV and then later it was deleted, that's a violation of infinite sites because you lost it. So how do you deal with that? So that's some of the work that we do. And yeah, with single cell sequencing, as I mentioned, so it's very error prone, and like some of the newer high throughput technologies, they're extremely low coverage. So it's very sparse data. Can you make more sense out of it if you impose an evolutionary model? If you try to do tree inference on top of it. So those are some of the questions that we look at. Yeah, but I think this is actually good to realize. So it's not just point mutations. It's quite complex.
Yeah. So for the structural variants, you mentioned insertions, deletions. Are there anything besides those two? Or is it
There's also like you don't have to change the copy number. So the copy number is like how many copies do you see? So for normal cells, it will be 2, except for like the sex chromosomes. But you could have like copy neutral, like structural variants. So what do I mean by that? So you could have like a transposition where or a fusion of 2 different genes, where you get a gene fusion. And some tumors actually have that. Like I think a famous one is like the Philadelphia chromosome in AML, where you have like a fusion between 2 genes that actually are on different chromosomes. So it's really 2 different chromosomes that are rearranged and then fused, and then you get this new 2 gene product.
So you have copy number aberration. Is that different from variation? Does it really have a different meaning, or is it the same?
Yeah. So copy number variation is typically used for germline, and copy number aberration (CNA) is used for somatic. So germline is you inherit from your parents. There's also variation that you get from your parents, and then typically use CMV for that. And the same thing for like SMV and SNP. So we don't use SNP because SNP is a population. And
the repeats, you can, there is some metrics on, certain percent of the human genome are repeats. Is that, so can you say something like that for the copy number aberration, or is that something it's different? You see what I'm saying? I know I've read papers where they say, okay, for example, in plants, they say, okay, this particular plant species has 80% repeats, right? So does that mean that it is 80% copy number variations, or are those two
copy number,
different?
yeah. Yeah, so copy number variation is like with respect to a reference genome. So if your reference genome, so essentially you need a reference. So for instance, if this is like my reference, like this whole piece of paper, and I start seeing like the same piece of paper, but like, you know, with like another half page extra, that's a copy number aberration, because you have like more material compared to what you had. And then maybe this extra quarter piece of paper comes actually from a duplication of this. It's always with respect to a reference. So the example that you gave of 80% repetitive, that's a statistic about like the reference genome. So, but a copy number aberration or variation is like, you know, you compare the reference to a new genome, like, you know, what's going on there. Is something of the reference replicated?
Okay, so then it's different. Then it's different.
Yeah.
Good. So my